Good day, Junior Techies, and welcome to this video looking at question 5 of the worksheet in our preparation for our exams. My name is Mr. P. I'm going to be working through this question and a few others with you just to help you better understand how to approach the questions and then hopefully give you a little bit of insight into some theory as well. Right, so let's have a look at question 5 here. So I tell all my students, and you would have heard me say this in previous videos as well, we got to get context from what we're given in the question first before we attempt to answer the questions in the actual question. So here we're given a diagram with 4 different diagrams. The context given to me in the heading here is that these represent phases of meiosis in an animal cell. All right, so we know the process that we're looking at here is meiosis, and I have diagrams 1, 2, 3, and 4. I can see that we have a few different labels there as well, A, B, and C, and I can try and identify those before we carry on. But let's jump into the questions here quickly. So I've now looked at my diagrams. I see I've got 4 diagrams, 1, 2, 3, and 4, each representing a different phase in meiosis. And here it asks me to identify the labels A, B, and C first. So this is a pretty simple question, a pretty straightforward question for me to answer first up. So let's have a look at these. If we look at A, we should have identified A as the centromere of the chromosome in the case of diagram 3. If we look at option B, or label B, we see that this is going to be the centriole, which is going to hold the spindle fiber pulling onto the centromere that we see in diagram 3, and then label C referring to or identifying the chromatid in this case, or we could refer to it as an unreplicated chromosome because the chromosome has actually been pulled apart by the spindle fibers. So I don't actually have a centromere that holds together 2 chromatids anymore. I actually have, in this diagram 3 now, an unreplicated chromosome that's moving to the pole of the cell in diagram 3. If we have a look at question 5.2, using the diagram numbers 1, 2, 3, and 4, place the diagrams in the correct sequence of meiosis. So in getting our context before, what we should have done is we should have tried to work out which of these diagrams would have had to have taken place first in the context of meiosis and then slotted the others in. So let's have a look quickly at these diagrams. If we look at identifying diagram 1, we can see that while we've got the invagination here of the 2 cells, we can see that I've still got chromatids joined by a centromere, so it can't be meiosis 2, but this has got to be the end of meiosis 1. So we can identify this as telophase 1. If we move on to diagram 2, we can see the moving apart of my chromosome, so the 2 chromatids still joined at a centromere moving to the poles. So we know this is anaphase 1. If it were to be meiosis 2, we would only see single chromosomes, not the 2 chromatids joined at a centromere. So diagram 2 we can identify as anaphase 1. If we move on to diagram 3, here we see those single chromosomes moving to opposite poles of the cell. So this is also going to be anaphase in diagram 3, but it's going to be part of meiosis 2. So we can identify it as anaphase 2. Diagram 4, we can see that these chromosomes are aligning themselves on either side of the equator in the middle of the cell. So here we've got metaphase 1. Now we simply have to put these phases in order. And we can see that if we look at PMAT that takes place twice, we can see that the order here is going to be 4, 2, 1, 3. Because my metaphase happens before my anaphase, which happens before my telophase, which happens before the anaphase 2. Right, if we look at question 5.3, how many chromosomes were present in the cell before meiosis began? I would go to the earliest of my cells. In this case, I'm looking at diagram 4, and I would count the centromeres. Because here we're looking at chromosomes and not chromatids, I would simply count the centromeres, and I would end up with 4 centromeres, which means my original cell would have had 4 chromosomes before meiosis began. Right, Junior Tukkies, I hope that this video has helped in an understanding on how to approach a question like this, because those diagrams 1, 2, 3, and 4 could be quite confusing if we don't get the context first. So approaching the diagram and getting an understanding of the diagram first does help when it comes to answering these questions. So, Junior Tukkies, thanks so much for joining me in this video. Please join me for further videos in this section. Get a hold of the worksheet as well as the worksheet memorandum and the theory booklet to help you prepare for this section in your final exam. Thanks so much, Junior Tukkies.